Okay, this lecture covers DNA and RNA structure and the following outcomes. We will revisit these outcomes at the end to make sure we covered everything. All right, so DNA and RNA are one of the four major types of macromolecules of the cell. You can see here, DNA makes up a very small portion of the chemicals in your cell. RNA makes up quite a bit more. RNA is a much more diverse molecule. There are tRNAs, there are mRNAs, there are rRNAs, and then there's a whole, I guess, another group of RNAs that we won't talk about, but if you get into molecular biology type classes, RNAs are really important in regulating gene expression and affecting how our cells work. So let's talk about these nucleic acids. So we're going to be talking about some of the differences between RNA and DNA. Some of the things that you can note from this image is that RNA is single-stranded. DNA is found in our cells double-stranded. And this structure, this kind of twisty two strands, oops, did I draw the same strand? No. Okay, two strands, we'll see it later, is called a double helix. So a helix means kind of like a spiral, like a slinky, and so this is a double helix, kind of two spirals intertwined. Okay. What I want you to notice is that they both have nitrogenous bases, which are these AGCT and AGCU, and they're called nitrogenous base because of all the nitrogens. So that's a similarity. They both have what we call a sugar phosphate backbone. So this ribbon here is made up of repeating units of nucleotides, which is made up of sugar and phosphate on the backbone, this outside part, and then all of these little lines sticking out represent the nitrogenous bases. There are some differences, such as RNA uses the base U, DNA uses the base T. Okay. So we'll talk about this with transcription, and you'll see how base pairing works, and that normally A and T base pair, but between RNA and DNA, A in DNA base pairs with U in RNA. Okay, so I'm giving you a lot of information right now. We're going to come back to it, but I think it's helpful to hear it a couple different times. I think that's really all we can see from this diagram. So let's focus on DNA first. DNA, the name, deoxyribonucleic acid, right? So DNA. And the deoxy stands for the sugar. Uh-oh. Okay. And you're going to see that it's called deoxy because there is no oxygen with this hydrogen. Deoxy, lost oxygen. Okay, so I'm going to erase that. There's no oxygen there. Okay. Nucleotides are the building blocks of DNA. We've talked about that. So nucleotides are the monomers. And we've talked about what it's composed of, three parts. A phosphate group, a pentose sugar, which in this case is deoxy. So this is different than glucose. Remember, glucose kind of looks like that. This is a pentose, so it's a 5-carbon sugar. And those carbons are named, or numbered, I should say. So I want you to see that it's 1 prime, this is a carbon, 2 prime, 3 prime, this is the 4 prime carbon, this is the 5 prime carbon. Okay, this is going to be really important when we talk about the 5 prime and the 3 prime end of a DNA strand. And it's named after the carbons of ribose. Okay. And then it has a nitrogenous base here, which we'll talk about in a minute. It can be the A, G, C, or T, and there are actually 2 types of nitrogenous bases. Let me make sure I'm covering everything. Deoxy is the sugar, phosphate is on the 5 prime carbon, so you're going to see that we call the phosphate the 5 prime end. Okay. Your nitrogenous base is here, and 3 prime end is going to be super important because that's where we hook the next nucleic acid. Alright, so you've got your 3 parts. Phosphate, sugar, base. So a lot of times I just draw DNA or RNA like this. Phosphate, sugar, and a base. The base can be different. And that is shown right here. You need to know the difference between purines and pyrimidines. Okay. So purines are bases with 2 rings. So you see 1, 2. So the way I remember it is purines have the small name, big structure. Because you're going to have to be able to identify different nucleotides and name them and tell me if they have adenine, guanine, thymine, or cytosine. So how do you remember which are purines and which are pyrimidines? So one student told me they remembered that the purines were A and G because this class was like pure agony, which I actually thought was funny. I didn't get offended. Okay, so purines, A and G. And then you just have to know pyrimidines are the other, T and C. So they have the big name, but the small structure. Okay, and when we talk about RNA, you'll see that uracil, which is the RNA nucleotide, sorry I get something, looks very much like thymine. Okay, so we represent these just by the single letter, A, G, C, T. Okay. So let's look at all these different nucleotides. Again, purines, small name, big structure, pyrimidines, big name, little structure, pure agony. Okay. Notice these all have phosphate, sugar, and then a base. So here's where you get your phosphate sugar backbone by connecting nucleotides. All of these bases have a 3' OH. The 3' OH is where the next nucleotide is added. So where we make our phosphodiester bond. You might also notice that these nucleotides have 3 phosphates. So in fact, ATP, our energy molecule, is an RNA nucleotide. So when we talk about how we put DNA together, these molecules already have their own energy. All right. DNA is a double helix, so it's like a twisted ladder. So now you can imagine this red ribbon as being the sugar phosphate backbone. And the two strands are held together by hydrogen bonds. And if you remember our old friend water, you get a partial charge on these molecules when they bind to oxygen or when you have nitrogen. Nitrogen usually has 3, right? And then between the slight positive and the slight oops. Oh, it didn't change, darn it. Between the slight positive and the slight negative charge, we make these weak hydrogen bonds. And we draw them as ... because to show they're weak, compared to a covalent bond, which is nice and strong. I shouldn't put that in this color. So that is a polar covalent bond. Okay, so you can see these are got ... dots all around them, and that's what's holding the two strands together. So it's by hydrogen bonds between two bases. Okay. So we'll look at that in a minute. And this is just a kind of a cute little video talking about the structure of DNA. You might want to watch it. It's only about 4 minutes. Okay, directionality. All right. DNA has 5' and 3' ends, and they're labeled after those sugars. The 5' phosphate is hooked to the 5' sugar, and the 3' OH, you can see, is where we connect. Well, you can't see that with red. Where we connect right? So we connect the 3' OH to the next phosphate. This, my friends, is your phosphodiester bond. Okay. So when you're looking at a structure of DNA, you always look for the phosphate, that is the 5' end, and you look for the lone sugar, that is the 3' end. What I want you to see now is that the other strand is what we call anti-parallel. Right? So it's kind of upside down. You have the 5' end here and the 3' end up here. So we talk about the directionality of DNA and RNA being 5' to 3', and the two DNA strands are anti-parallel. The other thing I want you to see is the base pairing. Okay, so you've got to remember that base pairing rules, that A base pairs with T and G base pairs with C. And I want you to notice that between A and T I drew 2 dots, and between G and C I drew 3 dots. Okay. That's because A and T make 2 sets of hydrogen bonds. But when G and C base pair together, you get 3 sets of hydrogen bonds. And it doesn't matter which strand G is on, it will always base pair with C, and T and A will always base pair. Okay, so they're always across from each other on our DNA sequence. So this is just showing you a little bit more. Hydrogen bonds are weak. We draw them as dot, dot, dots. But when you have lots of them, they can hold that DNA double strand together. It's important that we have weak hydrogen bonds because we want to be able to break these bonds. For DNA replication and for transcription. So you don't want to have really strong bonds holding your DNA strands together, or else you couldn't open up the two strands of DNA and replicate it. Alright, so we're looking at this again. This is just one strand of DNA. I like to show you lots of pictures so that you can see that the nucleotides are linked. Again, 5' here to the 3'. Okay, again, that's your phosphodiester bond, and you need to be able to recognize this, identify it. The phosphodiester bond is a covalent bond. So every time between a phosphate and a sugar, a phosphate and a sugar, a phosphate and a sugar. That's your phosphodiester bond. Okay. Now you can easily see sugar-phosphate backbone, right? It's repeating, and what's different is which bases are bound to the nucleotide. So let's talk about making those phosphodiester bonds. So we've talked about this before when we talked about the 4 types of macromolecules. We talked about dehydration synthesis and hydrolysis. So let's look at this one more time. Okay. Dehydration synthesis, anabolism, or an anabolic reaction. Remember, you lose one water molecule for every covalent bond you make. And remember that these little circles represents a nucleotide in this case, right? So you're hooking monomers, you're hooking nucleotides together, we abbreviate it NT, and you make a long polymer, you make your DNA or your RNA or your nucleic acid. Oh, one other thing. This is done in DNA by an enzyme called polymerase. Poly means many, mer is hooking them together, and ase means enzyme. So it's a many hooked together enzyme. Doesn't really make sense, but you understand. It's the enzyme that can hook many things together. Remember that when we break these strong covalent bonds, we use water. We do a hydrolysis. Okay. So for every bond broken requires or uses one molecule of water. Okay, so hydrolysis, just the opposite of dehydration synthesis. So let's look at this now specifically for DNA and RNA. So they're made the same way because they're hooking to nucleotides, right? And what happens is the 3' OH is now bound to the 5' phosphate. These two phosphates are lost. Okay, so this bond is broken. This blue bond is made. That's your phosphodiester bond. Here's your two phosphates floating away. So these could go, right? They're inorganic phosphate. These could go on and go into the mitochondria and be used by ATP synthase to make more ATP. So lots of stuff is recycled in the cell. This is your energy source. So in fact, when you take one nucleotide, let's see, nucleotide and hook it to another nucleotide, you actually release energy because they already have their own. So this is the one that kind of hurts your brain because it's anabolic and it requires energy, but they have high energy bonds, and so these molecules do not need extra energy added. These phosphodiester bonds are made during DNA replication, and during transcription by an enzyme called RNA polymerase. Okay, we're focusing on DNA, but I want you to know that this concept is exactly the same mechanism for RNA. Okay? It's always added at the 3' end for DNA and RNA. Let's write this. New nucleotide added at 3' end. So I want you to get in the habit of writing 5' to 3', and if you write it with that arrow, it will remind you that this is the way the chain is being synthesized. This is where the next monomer gets added at that 3' end. This is the way, direction the chain grows. Okay, so I think what I have next for you are a couple clickers to test, see how you're doing. So remember, just pause and then I will reveal the answer. Test yourself though. So name this nucleotide. Okay. So it forms 3 hydrogen bonds and it has a big structure. So big structure equals little name. That means it has to be a purine. Okay. And if it makes 3 hydrogen bonds, we know that A and T make 2, G and C. Here are your purines. Oops, I didn't spell that right. And here are your pyrimidines on this side. So if you get in the habit of writing things like I show you, it'll help you keep everything organized. So in fact, this is guanine. All right, try this one. Name this base pair. You've got to do it in the right order, left to right. Okay, so it has 2 hydrogen bonds, so I know it's either, I know it's not this, right? G and C have 3. Now I look and I say, okay, big structure, little name, has to be A or G. In this case it's A. This one is T, so the base pair is TA. All right, try this one. What describes a dehydration reaction? So the answer is C. Dehydration reaction is used to form polymers. Right? When we add water, or we break things down, or we form a nucleotide from DNA, that's breaking things down, those are all our hydrolysis reactions. All right, I really want you to try this one. You'll see, I'm sure, a question like this on quizzes. If DNA was composed of 10% thymine, what would be the percentage of guanine? So what I want you to remember is for every T, you have to have the same amount of A's because they base pair. And so we're talking about a double helix. So that equals 20%. G and C must equal 80% to get 100%. So then we have to divide it in half because we have the same number of G's as C's. So the answer is 40. Okay, let's finish up with a little bit about RNA. Okay. Strands of nucleic acids, what works for DNA works for RNA. So I think we did this chart back with the macromolecules. If not, I highly suggest you print it out. Test yourself. Look at this image and see if you can figure out the similarities and the differences. Remember, I said earlier, one of the big differences is the sugar. So RNA stands for ribonucleic acid. DNA, remember, stands for deoxy. And you can see that right here, right? RNA has 2 hydroxyl groups on the 2' and 3' sugar. DNA only has the 3' hydroxyl. It has lost the 2'. So they're both called nucleotides. They're both made of phosphate sugar base. The difference is here shown is the sugar. So you need to be able to recognize this, and what you do is you look for that 2' OH. So RNA nucleotides, just the same phosphate sugar base. The sugar is a little bit different, right? Has this extra OH group and one different base. So it has uracil instead of thymine, which is found in DNA. Okay. These are still purines. These are still the pyrimidines. So I said that RNA is a single-stranded molecule, but it can have very complex structures. So here are what tRNA molecules look like. Now we're going to draw them like this clover leaf because it's way easier to draw the clover leaf than to draw the actual 3D structure. And so tRNAs are super important in translation, making proteins by reading mRNA. This beautiful guy over here is ribosomal RNA from bacteria. This is a single strand of RNA. And let's see if I can remember, does it show the 5' and 3'? Okay, so here's one end. And 870, 970. Here is, oh, the other end, right? So it's this beautiful molecule that folds and folds and folds and folds and folds. Gives ribosomes, remember our protein-making machinery, structure and function. In fact, the RNA, the ribosomal RNA, is the enzyme that catalyzes peptide bonds in protein formation. We'll talk about that again when we could talk about translation, but the R stands for ribosome. T stands, I call it translation RNA. It's technically called, oh my gosh, I just forgot. What is the T? Ribosomal RNA and transfer. I was like, oh no. Transfer RNA. It's transferring, it's bringing an amino acid to the ribosome. Okay. And look at, it's, if you can see, I don't know if I can, I never can seem to zoom when I want to. But you can see that it follows base pairing rules. So G's and C's are base paired. Sometimes G's and U's base pair in RNA, that's different. And if you can look up here, A's and U's base pair as well. Okay, so again, I mentioned that we're going to talk about 3 types of RNA: mRNA, rRNA, and tRNA. But there are lots and lots of other types of RNA molecules. Okay, functions of DNA versus RNA. So DNA is our genetic material. It's kind of our storage. It's a very stable molecule. It's found in the nucleus of eukaryotes or in the nucleoid region of prokaryotes. The functions for RNA are transcription and translation. In eukaryotes, RNA is made in the nucleus but functions in the cytoplasm. At least the type of RNAs we're talking about. Okay, so made in the nucleus, functions in the cytoplasm, and its function is in transcription and translation. And again, we're talking about mRNA, tRNA, and ribosomal RNA. Okay, so I have some clickers for you. Go ahead and figure out what type of nucleotide this is. Okay, so you see 2 hydroxyl groups. This is RNA. Which arrow points to the phosphodiester bond? The answer is D. It's always between the sugar and the phosphate. Ooh, now look at this. Which is the 5' end of the nucleic acid? The answer is D, B, and C. Always look for that phosphate, right? Because here's the sugar that we're going to add a new nucleotide to. That's the 3' end. The 5' end is always where the phosphate is. And finally, take a look at this one. Is this DNA or RNA? And hopefully you noticed this OH here, that means it's RNA. If it was just a hydrogen, it would mean it was deoxy, it would mean it was DNA. Alright, so this is a nice little summary slide, how DNA and RNA are similar, yet different. Okay. Be able to talk about these differences and similarities, maybe in like a compare-contrast kind of thing. So let's see if we got all of this information. How dehydration chemistry is used to build monomers into polymers. We talked about that. And hydrolysis chemistry is used to break polymers into monomers. So that's all these guys, right? Dehydration synthesis and hydrolysis. Okay. Be able to identify the phosphodiester bond in DNA and RNA. We just went through that with the problem. Know the structure of a nucleotide in terms of the way in which the base and the phosphate group are attached to the sugar. Okay. So, phosphate group is on one side, the base is on the other side, the phosphate is at the 5' sugar and 5' carbon. The base is attached to the 1' carbon. Remember, those numbers are all based on the sugar, the ribose or the deoxyribose. Compare and contrast DNA and RNA structurally and functionally. So go ahead and fill out that chart to help you with that. Draw a schematic representation of a DNA and an RNA nucleotide and use it to compare and contrast the two structures. So this is as much detail as I expect you to be able to draw. Of course, you would add the OH if it was RNA, and if you had to draw it, you would just have an H if it was DNA. Further, for these bases, you should be able to draw like 2 rings for A or G and 1 ring for C or T. You don't have to know the whole entire structure. I don't even have that memorized. But a general schematic lets me know, you know, what nucleotides look like. Explain the base pairing rule of the DNA double helix. Who pairs with who? Well, now you know A and T pair, and they form 2 hydrogen bonds. And G and C form 3. So you can also just write it A, T, G, C. And why is hydrogen bonding important? Well, for DNA, it holds the 2 strands together. It holds our genetic material together. It keeps it safe, it keeps it stable, and then they're also important that they can be broken and copied. Explain what makes the DNA double helix anti-parallel. So that's this idea of 5' to 3', 5' to 3' going in opposite directions. And that's the only way the double helix will fit together. So the strands have to be anti-parallel. Describe directionality of a strand. That's 5' and 3'. Why is it called 5' and 3'? Remember, that has to do with the sugar, carbon numbers. So, practice drawing these nucleotides, remembering your purines. So let's put that here, your purines and your pyrimidines. Remember your base pairing rules. And that's going to be super important when we talk about DNA replication and then eventually transcription. All right, thank you.